Good morning, students. Welcome to the biology class. We are continuing with our same chapter, structure and the function of the cell, that is a unit of life. In prokaryotic cell, we have discussed about our outer covering, that is plasma membrane and cell wall. And today we will discuss nucleus and its structure. Some basic concepts of the nucleus. Nucleus was discovered by the English biologist Robert Brown in 1831. It is an important part of the cell, exerting a controlling influence on all the cell activities. It is usually spherical or oval structure, mostly located in the center of the cell, but in the plant cell they are at the periphery, that is eccentric. Some sieve tubes of the vascular plant, that is of the phloem, that is a vascular tissue. So phloem, that is having a sieve tube, sieve cell, companion cells. So sieve tube of the vascular plant and the red blood cells of the mammal lack nuclei at the maturity, means they are without the nucleus. They are enucleated cells. So the example of enucleated cell, that is your RBCs, and in plant, that is a sieve tube of a vascular plant. Nucleus is a membrane bound structure that contains the cell's growth and the reproduction. So it is a membrane bound structure. Eukaryotic cell is having a nucleus which is being surrounded by the membrane, and it is also being known as a true nucleus. It is commonly the most prominent organelle in the cell. The nucleus is surrounded by the structure called as a nuclear membrane. The membrane separates the contents of the nucleus from the cytoplasm. In this way, we have divided our cell into three compartments, that is outer covering, in which we discuss plasma membrane and cell wall, inner, that is your nucleus, and in last we will discuss about the cytoplasm, which is the ground substance along with the organelles. So structure of nucleus. An interphase nucleus. What is the meaning of interphase? When nucleus is at the rest, not dividing. It shows the following part, that is your nuclear membrane, nucleoplasm, nucleolus and chromatin reticulum. In this image you can see this is your outer membrane. This one, this is your outer membrane. This is called as a nuclear membrane. You can see that is of double membrane, outer and inner, and in between there is a space which is called as the perinuclear space, and the outer membrane is being studied by some red dot like structure which are being known as the ribosomes, along with this network which is being known as the endoplasmic reticulum. Inner to the membrane, and along with that you can see some gaps over there. These are some perforations. These are known as a nuclear pore. This is having a very important function, that how the outer cytoplasm substances connected with the nucleoplasm through these pores. So this is the nuclear membrane, which is double membrane layer, outer membrane, inner membrane. Along with there is a space which is called as a perinuclear space. They are having a ribosomes and the reticulum. Inner they are having the ground substance which is called as the nucleoplasm, and you can see these thread like structures are there and one dot like structure. This dot like structure is the nucleolus, and the thread like structure which is completely being embedded in the nucleoplasm, that is known as the chromatin material, or you can say that is a chromatin reticulum. So first, that is your nuclear membrane. Each eukaryotic nucleus has double membrane boundary consisting of protein and lipids with an outer and inner membrane. So the composition of double membrane is same as that of a plasma membrane. The two membranes are parallel with the space in between them called as perinuclear space. outer membrane is more often continuous with endoplasmic reticulum and is studded with ribosome, while the inner membrane is smooth. The nuclear membrane is interrupted at the several places by the minute pores which are formed by fusion of its two membranes. So this is the structure of the nuclear membrane, or you can say the detail of the nuclear membrane, that it is double membrane with outer and inner membrane, and the membrane they are having a space in between them which is called as a perinuclear space. Outer membrane is continuous with endoplasmic reticulum, which is a network like organelle. Along with them they are having a ribosomes which are being embedded or studied with them, whereas inner surface is smooth. A nuclear membrane is interrupted at several places by the minute pores which are being known as your nuclear pores. So what are nuclear pores and its function? That is your NCERT question, very important question it is. Many pores perforate the nuclear envelope or the nuclear membrane, and these are called nuclear pores. Function: these pores are passage through which the movement of RNA protein takes place in both the direction between the nucleus and the cytoplasm. So this is a very important function of your nuclear pores. Next is your nucleoplasm, that is your part of the protoplasm found inside the nuclear membrane, and another name of your nucleoplasm is nuclear matrix. It is a ground substance of the nucleus which is transparent and semi-fluid, and it contains the chromatin, reticulum and nuclei. Next is nucleolus. One nucleolus and two nuclei. So nucleolus is in a singular form. More than one nucleolus is present, then it is being known as a nuclei. Is found in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm because they are not having any membrane. So they lack the membrane. So content is along with the nucleoplasm. Nuclei are larger in those cells which are actively engaged in protein synthesis, and the nuclei are formed by nucleolar organizer region of one or two chromosomes. What is the main function of your nuclei? That is a site of active synthesis of ribosomal RNA. They make ribosomes, so they help in the protein synthesis. So very important question, what is the function of the nucleolus? Means they helps in the synthesis of a rRNA, or which particular part of the nucleus is responsible for the formation or the synthesis of RNA? The answer is nucleolus. And there is also one very important feature of the nucleolus, that is they are without the membrane and they are lying as such in the nucleoplasm. Next is your chromatin reticulum, that is the network of nucleoprotein fibers lying in the nucleoplasm, that is chromatin. Chromatin, they contains DNA, some basic proteins known as histones, and non-histone proteins and RNA. So this is the chemical composition of your chromatin. A single human cell has approximately 2-meter long fiber of DNA, that is chromatin, distributed among all the 46 chromosomes, because human being they are having a 46 chromosome, or you can say 23 pairs of the chromosomes. When cells start division of chromatin, the chromatin condenses into individual rod-like structures called chromosomes. Because we are discussing about the interphase nucleus, means when the cell is not dividing, it is not undergoing the cell division. At that time there is a network of the chromatin is there, but when the cells start division, that chromatin they become short and condensed to form the structure known as your chromosomes. So chromosomes we will discuss separately, and till now we have discussed about the nucleus. If the question comes, describe the nucleus along with the diagram and the function. So you have to give the four parts of the nucleus, that is about your nuclear membrane. Then you have to discuss about the nuclear pore. Then you have to discuss about the nucleoloplasm, then nucleolus and chromatin reticulum. Chromosomes. Chromosomes are thread-like structure usually present in the nucleus and become visible only during the cell division. As we have discussed, when the cells start division, the chromatin material, they become short and condensed to form the rod-like structure which is being known as the chromosomes. So they contain the hereditary information of the cell because they are made up of a DNA and protein, and DNA is known as the genetic material. In eukaryote, chromosomes can only be seen during the cell division, and that stage is known as the meta stage. So this is the structure. In this image you can see the structure of the chromosome. You can see it is a rod-like structure, consists of the arms. These are known as the chromatids. So these short long arms, that is being known as the chromatids, and they are being attached at one particular point in the center which is known as the centromere, or you can say this is a primary constriction which divided the chromatin into two parts, and depending upon the position of the centromere we are having a different type of chromosomes. So the structure of the chromosome, they are having the arm-like structure, that is your chromatin. In the center you are having a constriction, that is your centromere, and they are made up of DNA and proteins. So each chromosome consists of two arms, that is known as the chromatids. Every chromosome has primary constriction or centromere, non-stained region that holds the chromatin together. The chromosome become attached to the spindle fibers by its centromere. So disc-shaped structure called as kinetochores are present on the side of the centromere, which make them to be hold on the spindle fibers, that is during the cell division. A few chromosomes have non-staining secondary constriction at the particular location. This gives the appearance of small fragments called satellite, and each chromosome are called sat chromosome. Each of such chromosomes are known as your sat chromosomes. In this image you can see these are the chromatin. This is your primary constriction, that is your centromere, and this is your secondary constriction which is known as your satellite, and such chromosomes are known as the sat chromosome. So the chromosome showing the satellite region with the secondary constriction known as your sat chromosomes. Here you can see the types of the chromosome depending upon the position of centromere. So if the centromere is in the center and both the arms, that is the chromatin, they are of equal length, such type of chromosomes are known as metacentric. If they are being shifted a little bit towards one side, so upper one is shorter and lower one, they are the longer, means they are of the unequal, then it is submetacentric. In this you can see the one they are being reached to the poles, or you can say towards one end, and you can having a long chromatid on one side only and very short on another side. Such type of chromosomes, they are known as the acrocentric. But if we are having a centromere at the center only, but there is no chromatin at one pole and another they are having, that is in the telocentric, and they are not present in the human beings. So we are having a four types of chromosomes depending upon the position of the centromere. What is centromere? It is a primary constriction which separates the chromatids. They also make them to be hold at one position during the cell division, that is with the spindle fibers. So types of chromosomes depending upon the position of centromere, there are four types. Telocentric, the centromere is terminal. Terminal means at the end. and so the chromosome has only one arm. Metacentric centromere is right in the middle and two arms of the chromosomes are equal in length. Submetacentric, the centromere is little away from the center and so one arm is slightly longer than the other. Acrocentric, centromere is situated closer to one end, making one arm extremely short and another arm very long. So depending upon the position, we are having four types of the chromosomes. That is telocentric, having the chromosome has only one arm. They are having equal arms, and that is having a longer in one and slightly to the others, and they are having the unequal arms. So this is all about our nucleus along with the chromosomes. Very important question. We used to come 5-mark question we can have, that you have to give the detailed microscopic structure of the nucleus during the interphase. If the word come interphase, don't get confused. It means the nucleus at the resting position when you have to explain about the chromatin reticulum. So along with the diagrams, a very simple diagram I have given to you when we have discussed about the structure of the chromosome. You have to draw this diagram along with the detailed structure of the nucleus. Functions. It is essential for the survival and the long-term continuation of the cell. It maintains all the metabolic activities of the cell, cell maintenance and the cell replication. That's why it is also being known as the director of the cell. It regulates inheritance of characters from generation to generation because they are having chromosomes which are made up of a DNA and protein, and DNA is a genetic material which carries the information from one generation to the next generation. It controls the synthesis and regulation of activity of the enzyme. So these are the important functions. So chromosomes, or you can say the function of the nucleus, they are at the same one. So this is also being known as the function of nucleus. Instead of simply we can say that is the function of the chromosome. So in this you can say the function of nucleus oblique chromosomes. So the next lesson we will discuss about the cytoplasm and its organelles, in which we discuss each and every organelle one by one. For example, we are having the endomembrane system, then we are having a mitochondria, we are having a plastids, ribosomes. So one by one we will discuss, and till then, stay home, stay safe, stay healthy. Goodbye. Thank you.